at National Institute of Virology, which is right next door. It's almost a similar kind of a building, but of course, there's no Pavanam Malhotra there, so they're not as good. So this needs introduction. We work on Plasmodium and typically in the blood stages. We also have been working on Toxoplasma, and Cyril already introduced. And as he highlighted, because it's an extremely good model for studying and you can do things easy, such as genetic information. It can make better pictures. And it's very difficult to take students off. They start working on it, then they don't want to work on Plasmodium. So although we have done a few bit for last few years, but then we have tapered down on the Toxoplasma. Kinases and phosphatases cause reversible phosphorylation, and that basically is important for almost all signaling events, all processes in most cells. So it's a cliche, but it's a fact. And then Plasmodium has around 80 to 90 protein kinases, and then as shown, I have to mention from Vita's paper, around 27 phosphatases, and so on and so forth. And many of these are essential. And likely to be essential in all kinds of fatty acid available these things, so on and so forth. But then what has been very important and what we're trying to do, if you can stop that, please. I'm not used to limelight. Is to understand how these signaling pathways are organized in the parasite, and then which is clearly important. The idea was when we started out almost 2 decades back, is to the dream was to get a know-how of signaling maps in the parasite like we have in mammalian cells or in yeast and so on and so forth. This is just a snapshot of last 10-15 years of work, where we actually just shows our interest in second messenger signaling, such as calcium or phosphoinositides, how they regulate processes in the parasite and by what mechanisms. So many students, many collaborators have contributed to this simplistic picture and so on and so forth. I'll just show 2 brief examples of these and so on and so forth. For instance, we showed that calcium is involved in invasion regulating kinases such as these, calcium-dependent protein kinase 1, PKB, phosphoinositides regulate hemoglobin trafficking via proteins like FCP or epicoplasm biogenesis by ATG18 and ATG8, and CDPK7, which is another called calcium-dependent protein kinase, but is actually an effector of phosphoinositides, regulates parasite division and so on and so forth. This is just a brief stopover. I mean, we showed several years back, which showed where we were actually looking at effectors of calcium signaling in the parasite. We had actually shown prior to this study that phospholipase-mediated calcium signaling is important for host cell invasion. And then that's where we started working on calcium effectors such as calcium-dependent protein kinase 1. We showed that it crosstalk with the cyclic AMP signaling and regulates proteins like EBA175 and the secretion, and also regulate protein ROPHT, which we collaborated with Pavan to show that involved in the process of invasion. I had to throw this in to qualify this talk because it has a French connection. Many years back, we have shown that Plasmodium CDPK7, which is a effector of phosphoinositides, is important for parasite development. And then Wasim Daher had followed it up in Toxoplasma, that he also showed that the Toxoplasma homolog replication. So when I collaborated with my very important collaborator, Keshav Prasad, trying to do comparative phosphoproteomics to identify the targets, we stumbled on proteins which are involved in something which we hated at that point in time, phospholipid biogenesis, clearly because we did not know much about phospholipids, and we may know a little bit more courtesy Cyril Bote. And I think it's precisely this kind of a meeting, I think 4 years back, where we presented some low resolution TLCs to show that something in phospholipid biogenesis is going on with the mutants, which we had generated both in Plasmodium as well as Toxoplasma. So Cyril came to our lab and he said, we can do lipidomics for it. And then the effort of those resulted in 2 papers where we actually went on to show the CDPK7 and in both these parasites regulate phospholipid biogenesis, except it regulates different phospholipids, phosphatidylcholine in the case of Plasmodium and phosphoryethanolamine in Toxoplasma. And it was very, very interesting to note, despite the same kinase of the this thing is regulating. Actually, it was not surprising The reason for that is the targets which we identified in the two parasites of phosphoproteomics were different because the phosphorylation sites for kinase in toxoplasma was not present on the Plasmodium homolog and vice versa, and that actually was very interesting, and so this is where Cyril and we could on the lipidomics and so on and so forth. Coming to the central subject of today's talk, this is a genome-wide knockout study trying to delete all kinases done by a good friend Christian Doric, and then this is the essential kinome, the cis kinase. The attempts to knock out these listed kinases didn't result in parasites, or so suggesting that they may be essential. So we got interested in PK2 because it belonged to the calcium calmodulin dependent protein kinase family, and there's a couple of other reasons which I don't have time to get into. And then there was this study from Cato et al. Which showed that calcium calmodulin can regulate in vitro the activity of protein kinase 2. So given our interest in calcium signaling, we thought we'd like to explore that. But what turned out was then we express this enzyme in bacteria and do almost similar kind of experiments. So this is histone phosphorylation showing the activity of the kinase. Addition of calcium or calmodulin with or without the kinase did not matter. So it was independent, at least in vitro. It was not regulated by calcium calmodulin, while it was disappointing from that standpoint, but still interesting because it's an important enzyme. In addition to its kinase domain, as you would expect, it has a non-C-terminal domain, which is phosphorylated in the parasite, shown by the proteomics data by several groups. And if you need deletion of the C-terminal tail, it turns out it is critical for the activity of the enzyme. So this is the active enzyme, and if you remove this, what we call a putative regulatory domain as classified with this report, C-terminal domain, and you keep clipping it off and you get inactive enzyme, and so it is clearly critical, and we think it's important for the phosphorylation, the autophosphorylation of this domain may be critical for activity. Since we do not have the crystal structure and the alpha fold does not show very reliable structural details, so there's something interesting we'd like to explore. Now, since it is perceived or is an essential gene, we resorted to doing conditional knockdown, which is using standards approaches. In this case, we use a dimerizable recombinase, where rapamycin can bring the two active site centers of the rapamycin of the recombinase. And if you insert LOXP sites into the PK2 locus, you can excise this by providing rapamycin. So this is what all we did. A few years of work by student, because it was the first daiquiri line we made in our lab, and we checked imbalances by using sequencing and genotyping, and just to show if you add rapamycin, the LOX site can be removed and you have excision as shown here. And then of course the protein is significantly depleted when you provide rapamycin. So whenever I say R or RAP, that means that is a knockdown condition, and D stands for DMSO, so on, which is the control. There's an interesting localization, except we do not know where precisely it is localized. That makes it interesting because we tried. So it has these punctate structures present in the schizons, and we tried to look at the usual suspect, which is the rhoptries and the micronemes, and it did not, despite our best efforts to see it localize either of them. At best, what we could see was it is proximal to the micronemes, and that's as proximal as I'll get in my definition of its localization, and so on and so forth. And as you can appreciate here, this is just a zoom, but it is present either as one or two puncti inside the merozoites, and which could be interesting locations, and we do not know.
Typical merozoites are.
Sorry, asterisk. Remember that. Yeah, yeah. So we can talk. Yeah. So, like what we do is we do a growth rate assay. And it was very evident when you got these transgenic lines. When you provided rapamycin, they were growing very slowly, and as shown here, as this growth rate assays, and so on and so forth. And rapamycin was added and looked at the growth rate of the parasite. One of the first things which we observed was the parasitemia really dipped after 48 hours, which is entering into the next cycle. And what we saw was a lot of these merozoites, which were in the Jim smear, and I'll elaborate more on it, just sticking to the parasite and so on and so forth, but clearly they're not able to invade. But in addition, in a subset population of the parasites where they actually probably were successful, they were pycnotic abnormal structures such as these, and you could quantitate those from the Jim smears, and also shown here. This and so they were not transiting from rim to trophozoite, they were mostly pycnotic or abnormal morphologies and so on and so forth. And that is the last bit on this part. But what we were really interested is that what is happening here. Does it play a role in post invasion? And that is what and there was no defect in parasite division and all other things which we did. So what we did was a typical assay where you took the schizons and you put them in culture, and after a while, you provide them the new red blood cells, and they will go on to invade. They will egress out, burst out, and you will see the rings, which will be indicative of invasion. And we did it in more than one ways. To cut a long story short, there was no defect in egress. So this is all the line curves for the schizons, but the ring was significantly reduced post invasion when you in the rapamycin treated parasites, shown in green, compared to DMSO control. And then what we also did was we, in the meantime, we made a line where we provided another extra episomal copy of HA-TAC PK2 and so on and so forth. In this case, we could complement, as shown here, most of the defects in invasion. So the effects are real. Often we are not able to make complementation plasmodium, but in this case, we were lucky and so on. So the process of invasion, as most in this audience would know, is a complex but well orchestrated process where a merozoite goes and first has initial contact with the RBC, then it reorients and takes a business end, which is containing the apical end, the microneedles and the rock trees, which will secrete ligands, which will bind to the receptors on the RBC surface. It will invaginate the RBC surface. It will make something called what is called the tight junction. You will have the actin myosin motor, which is which will provide the force to enter the parasite, held together by the something called a glidosome, and so on and so forth. So we wanted to see which stages of invasion were affected. Clearly, we did an assay with cytokinesin D, which actually prevents invasion, but does not prevent attachment, and we did not find any defects in attachment. So then we went on to see what if there were subsequent steps in invasion. So we resorted to like microscopy. As you can see in the normal parasite, it is egress fine. It will go and attach and orient and do its job, and then modify the RBC membrane. And after a while, it will undergo ichnocytosis, and the RBC will recover after a bit. And there's another example of a normal parasite. And it will go on, and as you are watching this successful invasion going on here, I'll switch on the videos for the so for the depleted parasite or PK2 depleted parasite. Just focus your attention to this guy. It will attach. It'll keep making efforts to enter the RBC, and believe me, it will not be successful. Otherwise, I wouldn't show this event, right? And there's yet another example. So of another merozoite trying to invade and making unsuccessful attempts in doing so. You can make this and you can analyze the snapshots from these and quantitate these events. To cut a very long story short, we found was the attachment, as I said, was not affected. The parasites had failed invasion, and they exhibited prolonged, some of them showed prolonged attachment and also prolonged ichnocytosis, basically suggesting that it is the latter stages of the process of invasion, wherein they have made the contact, everything's gone on fine, but they are not able to breach the RBC membrane and make a vacuole out of it. So and so on and so forth. So there could be issues related to things like and those who know in the field, this was very typical of defects seen in a process called just tight junction or the moving junction formation. And that is what we set out to address. As I said, okay, this you already know. So this is just a sequence of events, and this is where something is, we feel none of these processes seem to be altered by several video microscopy images we analyzed, and it is probably somewhere here we see the defects may lie. So as I mentioned, the process invasion relies on proteins secreted from micronemes and ruptries. So we analyzed, and I'm just providing summary data for this, ruptry microneme proteins secreted by PK2 depleted parasites compared with the control, and turned out that microneme proteins AMA1 and EB175, as to show an example, the secretion or shedding from the merozoite was significantly impaired, where the ruptry proteins, the ones we analyzed, did not change. And so what we did was, AMA1 is a critical protein, and it plays an important role in tight junction formation because it makes a complex with ruptry neck protein, which is critical for making the tight junction, and then which is, as I said, critical for the parasite entry. What it undergoes several steps of processing while it resides in these punctate micronemes, as a parasite, as this process of regress and this thing matures, it comes onto the surface of the parasite as shown here, and co-staining with MSP1. This is already well known. What we observed was, in the case of PK2 depleted parasite, most of the AMA1 remain in the ruptries. It did not come to the surface, which typically is the case before it actually shed out of the parasite. And we have done quantitations of this and so on and so forth. So now next what we wanted to do was, what is the status of AMA1 at the tight junction? So you can actually stabilize the tight junction using cytochalasin D, which inhibits the polymerization of actin, and it will prevent invasion, as shown by many people, including this paper from where this schematic is taken. So AMA1 will be in these bead-like structures at the junction, as you can see here, along with RON2, and this is where it is. And since you have provided cytochalasin D, it is basically stuck at that this thing. In the case of normal DMSO treated normal parasite, the PK2 is intact. But what happens when you deplete PK2 by providing ribamycin? Most of these parasites, and we have done quantifications, AMA1 staining at the tight junction was not present. And given that AMA1 is pivotal and critical for tight junction formation, which I showed and as also shown earlier, is critical for the successful process of invasion. We feel this is a defect which is being caused, and as a result of this, we have PK2 depletion impairs the process of invasion. So there are many reasons why we are slow in our progress, and me being lazy, and the students are never lazy, so and so on. But also because we try to dig deep into identify mechanisms, and more often than not, it leads to frustration, so on and so forth. But then we still have make attempts. If we don't do that, then we will not carve out the pathways and so on and so forth. So what we did was, with Keshav and this student Neelam, we did comparative phosphoproteomics, where we compared the global phosphoproteome of PK2 normal and PK2 depleted parasites, and looked at differentially phosphorylated protein, which basically told you they were direct or indirect targets of PK2. Don't look too hard. I have a table to show you the good.
I see it. I've seen what I needed.
Yeah, you go blind with that. Yeah. The point is that two major classes of proteins, when we do string analysis or even cherry-picked things, which were altered, were either involved in cell signaling or involved in cell signaling, or proteins involved in glidosome assembly or actin myosin motor. And both of these are, as I said, are critical for invasion. And this is where I listed our key putative targets. Yes, you can say we say cherry picking, and then who doesn't do it, right, Cyril? So in this, the reason, and there was for good reasons, these proteins are, as I said, signaling proteins, and some of them and glidosome proteins, which have been demonstrated to have a role in invasion. So it made perfect sense to go and look for it. Especially the ones which caught our attention were these proteins, which are actually critical for cell signaling. Guanylyl cyclase, GC, has been shown, the cyclic gene, which is important for synthesis of cyclic GNP, has been shown by David Baker, Mike Blackman, others, important for egress invasion and it regulates calcium release. PI4P kinase, diacylglycerol kinase, the same thing, and so on and so forth. So you wanted to see how does PK2 regulate the process of invasion. So first and foremost, we looked at granulate cyclase alpha, with David Baker in a very elegant set of studies has shown that is involved in egress and vision. And it is a very different kind of GC, because it also, in addition to the cyclase domain, it has the Cyril's favorite P4 ATPase domain, which is important for a variety of things. And David has shown that in the nucleotide binding domain, that if you make mutation, actually it has an impact on GC formation. So clearly the ATPase domain, and the reason I'm bringing it up because of the phosphorylation site, which is putative PK2 phosphorylation site, or what was regulated by PK2, the parasite which we identified to be significantly hypophosphorylated resides in this domain, and so on and so forth. So to cut a long story short, does it have if that is true, does it have is it regulating GC? Does it have a bearing on cyclic GMP synthesis? The answer is yes. So when you deplete PK2, then the cyclic GMP synthesis is impaired in the parasite. And what David and others have shown, many others have shown, is that cyclic GMP signaling is critical, and via protein kinase G is critical, and it regulates calcium release from the parasite, and which is, as I mentioned, me and others, Chetan here and Abhishek is here and so on and so forth, is important for invasion. So we looked at, if you deplete PK2, what happens to calcium, the intracellular calcium level. So basically you can use A23187, where you basically essentially all calcium in the cytosol you can release and calculate. So the total calcium levels did not change the parasite upon PK2 depletion, but when you deplete, when you provide zephrin-NAS, it will stabilize phosphodiesterase and stabilize cyclic GMP formation, and deplete PK2 and look at intracellular release of calcium. There was significant depletion upon PK2 depletion. So basically, that brings me to the summary slide, since the organizer is interested that I don't have to speak too much. So this way, I think this is what we are going on. This is the model. We think we know PK2 is regulating microneme secretion and of proteins like gamma-1, and which is important for tight junction formation and invasion. And it may be achieving this by regulating cyclic GMP formation via GC alpha phosphorylation, which in turn, as shown by others by PKG, is important for PKG, cyclic GMP-driven protein kinase, which is almost the only target of cyclic GMP, a major target during the course of invasion. And then in turn, not being shown, but it's being speculated, and we have reasons to speculate that it may regulate phospholipase C, and which is extremely critical for calcium release, and so on and so forth. So the most of the work I described today was initiated by Ankit Gupta, now a faculty at AIIMS Raebareli, and majority of work done today was Rahul Singh Rawat, and we're followed by Monica, the lab, and these are the people who have contributed to some of the other studies. And Keshav has been a very important collaborator in genomics. Cyril has been, and Yoshiki first, and then Cyril, important for all the lipidomics work, which was extremely critical. And Pawan and Asif have been former collaborators of many things. And I mean, I'd like to see DBT, DST, and also recently we got a grant for India Alliance with Rita and Keshav to work on, which is a major activity in the lab, looking at cell division and how cell division is regulated by reversal phosphorylation and so on and so forth. Thanks very much. Thank you.
Long live India and France friendship.